Anatomy of dicotyledonous and monocotyledonous plants and secondary growth. Dicotyledonous root. The internal tissue organization is as follows. Epidermis is the outermost layer. In the form of unicellular root hairs protrude many of the epidermal cells. Several layers of thin-walled parenchyma cells with intercellular spaces are present in the cortex. Endodermis is the innermost layer of the cortex. Without any intercellular spaces, it comprises a single layer of barrel-shaped cells. In the form of Casparian strips, the tangential as well as radial walls of the endodermal cells have a deposition of water-impermeable waxy material suberin. A few layers of thick-walled parenchymatous cells, referred to as pericycle, lie next to endodermis. During the secondary growth, initiation of lateral roots and vascular cambium takes place in these cells. The pith is small or inconspicuous. In between the xylem and the phloem lies the parenchymatous cells called conjunctive tissue. Usually 2 to 4 xylem and phloem patches are present. Later, in between the xylem and phloem develops a cambium ring. On the inner side of the endodermis, all tissues such as pericycle, vascular bundles and pith constitute the stele. Monocotyledonous root. In many respects, the anatomy of the monocot root is similar to the dicot root. Epidermis, cortex, endodermis, pericycle, vascular bundles and pith are present in a monocot root. Usually in the monocot root, more than 6 xylem bundles are present as compared to the dicot root which have fewer xylem bundles. A large and well-developed pith is present. Secondary growth does not take place in a monocotyledonous root. Dicotyledonous stem. A typical young dicotyledonous stem shows that the epidermis is the outermost protective layer of the stem in a transverse section. A dicotyledonous stem may bear trichomes and a few stomata and also cover with a thin layer of cuticle. The cortex consists of the cells arranged in multiple layers between epidermis and pericycle. 3 subzones are present in the cortex. A few layers of cholenchymatous cells are present just below the epidermis in the outer hypodermis, which provide mechanical strength to the young stem. Below hypodermis is the cortical layers. Rounded, thin-walled parenchymatous cells are present with conspicuous intercellular spaces. Endodermis is the innermost layer of the cortex. The cells present in the endodermis are rich in starch grains and this layer is also referred to as the starch sheath. On the inner side of the endodermis, pericycle is present and it also lies above the phloem in the form of semilunar patches of sclerenchyma. There are a few layers of radially placed parenchymatous cells in between the vascular bundles which constitute medullary rays. The vascular bundles are arranged in a ring in large numbers and this arrangement of vascular bundles in the ring is a characteristic of dicot stem. Each vascular bundle is with endarked protoxylem conjoined and open. The pith consists of a large number of rounded parenchymatous cells with large intercellular spaces which occupy the central portion of the stem. Monocotyledonous stem. A sclerenchymatous hypodermis, a large number of scattered vascular bundles, each surrounded by a sclerenchymatous bundle sheath, and a large conspicuous parenchymatous ground tissue is present in the monocot stem. Conjoined and closed vascular bundles are present in a monocotyledonous stem. Centrally located vascular bundles are larger when compared to the peripheral vascular bundles that are generally smaller. Water-containing cavities are present and the phloem parenchyma is absent within the vascular bundles. Dorsiventral leaf. Three main parts, namely epidermis, mesophyll, and vascular system, are seen through the lamina in the vertical section of a dorsiventral leaf. A conspicuous cuticle is present on the epidermis which covers both the upper surface and lower surface of the leaf. The stoma in the abaxial epidermis are generally present more in number than the adaxial epidermis. The adaxial epidermis may even lack stomata. The mesophyll is the tissue between the upper and the lower epidermis. Mesophyll is made up of parenchyma which possesses chloroplasts and carry out photosynthesis. The Palisade parenchyma and the Spongy parenchyma are the two types of cells in the mesophyll of the leaf. Palisade parenchyma, which is placed adaxially, is made up of elongated cells which are arranged vertically and parallel to each other. Spongy parenchyma, which are oval or round and loosely arranged, are situated below the palisade cells and extend to the lower epidermis. Between these cells there are numerous large spaces and air cavities. In the veins and the midrib, vascular system is present which includes vascular bundles. Dependent on the size of the veins are the size of the vascular bundles. In the reticulate venation of the dicot leaves, the veins vary in thickness. Surrounded by a layer of thick-walled bundle sheath cells are the vascular bundles. Isobilateral leaf. The dorsiventral leaf anatomy is similar to that of an isobilateral leaf in many ways. It shows the following characteristic differences. The stomata in an isobilateral leaf are present on both the surfaces of the epidermis, and the mesophyll is not differentiated into palisade and spongy parenchyma. Certain adaxial epidermal cells along the veins modify themselves into large empty colorless cells in grasses, and they are called bulliform cells. The leaf surface is exposed when the bulliform cells in the leaves have absorbed water and are turgid. Due to water stress, when they are flaccid, they make the leaves curl inwards to minimize water loss. As seen in vertical sections of the leaves, the parallel venation in monocot leaves is reflected in the near similar sizes of vascular bundles. Secondary growth. The primary growth is the growth of the root and stems and in length with the help of apical meristem. Apart from primary growth, most dicotyledonous plants exhibit an increase in girth, and this increase in girth is called the secondary growth. The tissues involved in secondary growth are vascular cambium and cork cambium, which are the two lateral meristems. Vascular cambium. Vascular cambium is a meristematic layer that is responsible for cutting off vascular tissues, xylem and phloem. It is present in patches as in single layer between the xylem and phloem in the young stem. It forms a complete ring later. Formation of cambial ring. The intrafascicular cambium in dicot stems are the cells of cambium present between primary xylem and primary phloem. Interfascicular cambium is formed when the cells of medullary rays adjoining these intrafascicular cambium become meristematic. Thus, a continuous cambium ring is formed. Activity of the cambial ring. Both towards the inner and the outer sides, the cambial ring becomes active and begins to cut off new cells. Towards the pith, the cells cut off and mature into secondary xylem, and towards periphery, the cells cut off and mature into secondary phloem. On the inner side than on the outer side, the cambium is generally more active. As a result, it soon forms a compact mass when the amount of secondary xylem produced is more than secondary phloem. Due to the continued formation and accumulation of secondary xylem, the primary and secondary phloems get crushed gradually. In or around the center, the primary xylem, however, remains more or less intact. The cambium forms a narrow band of parenchyma at some places which passes through the secondary xylem and the secondary phloem in the radial directions, and these are the secondary medullary rays. Spring wood and autumn wood. The activity of cambium is under the control of many environmental and physiological factors. The climatic conditions are not uniform through the year in temperate regions. Cambium is very active and produces a large number of xylary elements having vessels with wider cavities in the spring season. Spring wood or early wood is the wood formed during spring season. The cambium is less active and forms fewer xylary elements that have narrow vessels in winter, and this wood is called autumn wood or late wood. The autumn wood is darker and has a higher density, whereas the spring wood is lighter in color and has a lower density. The annual ring is of two kinds of woods that appear as alternate concentric rings. As seen in a cut stem, annual rings give the estimate of the age of the tree. Hardwood and sapwood. In old trees, due to the deposition of organic compounds like tannins, resins, oils, gums, aromatic substances, and essential oils in the central or innermost layer of the stem, the greater part of secondary xylem is dark brown. To the attacks of microorganisms and insects, these substances make it hard, durable, and resistant. It is called heartwood as this region comprises dead elements with highly lignified walls. The heartwood gives mechanical support to the stem but does not conduct water. Sapwood is a peripheral region of the secondary xylem and is lighter in color. The sapwood is involved in the conduction of water and minerals from root to leaf. Cork cambium. Due to the activity of vascular cambium, as the stem continues to increase in girth, the outer cortical and epidermis layers get broken and need to be replaced to provide new protective cell layers. Usually in the cortex region, sooner or later develops another meristematic tissue called cork cambium or phellogen, which is a couple of layers thick. Phellogen is made of narrow, thin-walled and nearly rectangular cells. On both sides, phellogen cuts off cells. The inner cells differentiate into secondary cortex or phelloderm, while the outer cells differentiate into cork or phellem. Due to suberin deposition in the cell walls, the cork is impervious to water. The secondary cortex cells are parenchymatous. Collectively, phellogen, phellem, and phelloderm are known as periderm. Pressure builds up on the remaining layers peripheral to phellogen, and ultimately these layers die and slog off due to the activity of the cork cambium. A non-technical term bark refers to all tissues exterior to the vascular cambium, therefore including secondary phloem. A number of tissue types refers to bark, namely periderm and secondary phloem. Early or soft bark is the bark that is formed early in the season. Late or hard bark is formed towards the end of the season. Instead of cork cells, the phylogen cuts off loosely arranged parenchymatous cells on the outer side at certain regions. Lens-shaped openings called lenticels are formed when these parenchymatous cells soon rupture the epidermis. Between the outer atmosphere and the internal tissue of the stem, lenticels permit the exchange of gases and they occur in most of the woody trees. Secondary growth in roots. The vascular cambium is completely secondary in origin in a dicot root. The vascular cambium originates from the tissue located just below the phloem bundles, a portion of pericycle tissue above the protoxylem forming a complete and continuous wavy ring which later becomes circular. To those already described above, further events are similar to for a dicotyledonous stem. Also, in stems and roots of gymnosperm, secondary growth occurs. However, in monocotyledons, secondary growth does not occur. Please share this video with your friends. Leave your comments below and please subscribe to my channel for more content. Click that bell notification that would let you know when a new video is uploaded. Thank you so much for watching. Once again, please subscribe to my channel for more content.